Hello everyone, welcome back. Today we are going to be talking about chapter 4, or the fourth objective, called biological molecules. Now, biological molecules are basically these very essential molecules or essential atoms, a group of atoms which made a molecule, and these molecules are very essential for life to exist. Now, one of these very biological molecules could be starch, for example, glucose, for example. So you will have to know the chemical elements which made up the carbohydrates, fats, and protein. Now, first of all, we have to talk about food. Why do we have to eat food every day? Now, we have to eat food every day to get energy and nutrients molecule from eating food, from chemical digestion. And food is mainly made up of three different parts. Now, the first one is carbohydrates, or you can call it carbs for short. And fats, it is made up of proteins as well. Now, these three are the main constituents which made up the food. Main constituent of food. Now, we have to talk about how these molecules are made. These carbohydrates, fat molecules, protein molecules, all made from smaller molecules such as monomers. Smaller molecules such as monomers. Now, these monomers could be various ones. So, for example, for protein, the monomers would be, the protein is a very huge macromolecule. It is a polymer, essentially. It is made up of different types of amino acids, right? So these amino acids are small subunits which join together and fold in complex 3D shape to produce protein. Now, there's an example for protein. Now, we'll have to know the different subunits from which all these molecules are made. Now, we have to look at starch, carbohydrates first. Now, starch, glycogen, and cellulose are made from glucose. Now, you will have to know the chemical formula for glucose. You don't really have to know the specific about glucose at this IGCSE level, but you will have to know its chemical formula. And this is 6,6H12O6. This is something that you have to remember as well. Now, glucose is really essential because, as you know, glucose is an energy molecule. In other words, we burn glucose with oxygen to produce carbon dioxide and water, and this in turn produces energy for us. Now, this reaction is the respiration reaction. So you have to know starch, glucose, glycogen, and cellulose are all made from the same subunits. These are the same subunits, or monomers. You can call them subunits, whatever you like. Right? Now, that's something that you have to know. Now, second of all, we have to know proteins are made out of amino acids. Now, I'm going to shorten this into using A.A. Now, this means amino acids. Right? Now, amino acids, you don't really have to know the structure of amino acids. You just have to know that proteins are made of amino acids. Okay. Thirdly, you have to know about fats and oils. Now, fats and oils are very essential for human beings as well. Now, how are they essential? Well, we know that fats help us insulate. These layers of fat help these different types of organisms to survive this cold, harsh winter, right? They help insulate, so keep our body warm Now an example of this would be in those seals, lion seals, or whales. Now they have something called blubber, and this is basically a layer of fat under the skin. Under their skin, this helps them to protect these seals and whales in the cold ocean waters. So yeah, and another function of fats and oil is basically that if you have learned about, you will learn it in year 11 next year in the junior second year of IGCSE course, but fats and oils actually help to insulate your nerve pathway, so insulate your myelin sheath, basically insulate your neurons pathway so that the electrical signal which your brain sends out will be faster, right, because it is insulated. Okay, now we have to know what fats and oil are made of. Now fats and oil they are made of two substances. They are made of fatty acids and glycerol. Now these two molecules, you have to draw them in a very basic structure. So we have to know that fats, mostly fats, or we can call them triglyceride. Remember, triglycerides and fats are mostly the same. In this level, most of the fats are triglyceride. Now these triglycerides are made out of 3 fatty acids plus 1 glycerol molecule. So the overall shape would be like this. Now the glycerol molecule is represented by this block, and 3 fatty acids are represented by these chains. Now in reality, these chains are really long because the carbon atoms are more than 15, around 50, 16, around something like that. So these chains of carbon are really long. Now this is the glycerol molecule. 1 glycerol molecule, we can see there are 3 fatty acids. Now you will have to draw this structure. Okay? Okay. Now, I do want to mention about something about starch, glucose, cellulose, that carbohydrates are also classified into different types. So there are 3 types. There are monosaccharides, there are disaccharides, and polysaccharides. Now polysaccharides are basically starch, glycogen, and cellulose, right? Because they are made up of many different glucose. Now poly meaning many of the same type, so it is basically a polymer itself. So polysaccharides. Now this example of polysaccharides should be cellulose, glycogen, starch, right? Now you have to know some of the examples for monosaccharides as well. Now monosaccharides, the best example is glucose, because glucose is the basic fundamental subunit in which cellulose and glycogen and starch are built up upon this molecule of glucose. Now disaccharides, you have to know there are 3 disaccharides. There are maltose, they are sucrose, and the last one being lactose. Now there are 2 more monosaccharides that I haven't mentioned. Glucose is one of them. Fructose, if you would like to call it that, is also another monosaccharide. And there is also one more which is called galactose. Now galactose is a sugar that is found, a sugar or carbohydrates. Now carbohydrates and sugars are often used interchangeably because they can be used in that way, because carbohydrates are basically sugar. Now saccharides, these root word saccharide mean sugar in Latin. Now poly meaning many sugar, di meaning made up of 2 sugars, and mono meaning made up of 1 sugar. Now you would also have to know where some of these sources of sugar come from. Now glucose definitely comes from tea bags, right? Sucrose also comes from those tea creamers and stuff that you put in your coffee to make it more slightly sweeter. Now fructose actually comes from those sweets in fruits, so such as apples, you can say pineapples, right, and many different kinds of fruits. They produce fructose. Now galactose and lactose are found in milk. Okay, so yeah, you will have to know some of the sources and also know that carbohydrates is actually classified into monosaccharides, disaccharides, and polysaccharides. Now, moving on, we'll have to also describe the test, the food test for each. Now, firstly, for starch, we are going to use an iodine solution. Now, if starch is present, this is called the positive result. If starch is present, it turns the iodine, which is solution, which is brown, dark brown in color, turns blue-black when starch is present. Positive result meaning starch is present, basically. Starch is present. Now, when starch is not present, negative result, this means it will stay the same color. The iodine will stay the same color because obviously there's going to be no change. Now, the second food test that you have to know is the Benedict's test, or the test for reducing sugar. Now, at this level, I should probably remind you that you only have to study reducing sugar. You will not have to study non-reducing sugars. Now, non-reducing sugar, I'm going off a bit of the IGCSE syllabus, but however, non-reducing sugars are sugars such as sucrose, lactose. These are some of the non-reducing sugars. And these non-reducing sugars have a different test from the reducing sugar. So, yeah, you don't have to pay this no mind. You will only have to focus on the reducing sugar test. Now, this reducing sugar test is called the Benedict's test. Now, this Benedict's meaning the scientists who actually came up with this test. Benedict's test. Okay. So Benedict's test is basically to add Benedict's solution. They add Benedict's solution to the sample of that contains this reducing sugar. Now, reducing sugar example could be glucose or fructose. These could be some of the examples of reducing sugar. Now, we add a small drop of Benedict's solution into the sample that you think contains this sugar. And if we add a few drops, right? If we add a few drops of sugar, the positive result, let's have a positive result. If sugar is present, if the reducing sugar is present, then it will turn brick red in presence of glucose. Now, note that this Benedict solution actually, if you try to do the experiment by yourself, you will see that Benedict solution itself in color. Color of Benedict solution is actually blue itself. Now, originally it was still blue. However, if you heat it, right, remember, we have to note another very important factor is that we have to heat the solution, right? We have this solution with Benedict solution, right? With Benedict's. Only then will the test work. If you don't heat it, then the color will still blue. Now, what actually happens is that a wide range of colors are shown. So first it will turn blue, then it might turn green, then it might turn orange, then it might turn it might turn red. Now, the end point of the final color has to be brick red. Now this actually depends on the concentration of glucose. Now I have not shown you all the color changes, but basically you will start from blue color and you will most likely end up with an orange or brick red color. Now Cambridge is really fussy about whether writing red or orange. They prefer using the word brick red. Okay, now the third test that I would like you to know is the test for protein. Now how do we test proteins? Well, we test proteins using a solution, yet again by another scientist, called the Burette test. Now the scientist's name was Burette. So he actually came up with this test. Now this Burette test, or you add a few drops of Burette's reagent, or you can call a few drops of Burette solution. Now if protein is present, positive result meaning protein is present, then the color will change from blue to purple or violet. Now the Burette solution itself, the reagent solution itself, the color of the reagent solution itself is blue in color. Now if protein is present, it will turn into purple or violet. Sometimes they do describe it as a violet halo color, halo ring. Now this is the same thing because in the test tube there's like a violet halo ring. Now it is called halo because it kind of looks like the angel, the circle things above angel. Right. So if the negative test, if protein is not present, then with the color obviously will stay the same. Now, a third test that I want you to know is the ethanol emulsion test. Now, this ethanol emulsion test is for fats and oils. Now, it is called ethanol emulsion test because we are using the reagent ethanol. We add a few drops of ethanol. Then we add a few drops of water H2O. We add a few drops of water. Secondly, so a few drops of ethanol is added, and a few drops of water is added. Now when you shake this ethanol and water inside the sample, that you know this is the sample. If you shake the test tube, holding the test tube with your thumb over the test tube, if you shake it really hard, you will see an observation. Now for observation, you have to see this. In the positive result, you will see a cloudy, almost like milky-like suspension forming. This means that the positive result means that the fats and oils are present in the sample which you added. Right. If it's negative, then the solution will remain clear because obviously the fats are not going to react with ethanol, and therefore they will not form any suspension or precipitate looking. Okay, and the last test that I want you to know about is the DCPIP test for vitamin C. Now, vitamin C, this is one of the newest, I think this was one of the newest changes to syllabus. So you have to know about the DCPIP test. Now, DCPIP test is basically, DCPIP is the reagent used to test for vitamin C. Now, this is actually a dye. DCPIP is a dye. So we are going to add a few drops of DCPIP test. Drop by drop, right? We are going to add. So the DCPIP test, right? As I was saying, DCPIP test is a dye, and this dye color is blue. The DCPIP reagent is blue in color itself. Now, it will turn pink in acid conditions. Basically, you don't really have to know most of what it does or the positive drops. However, you have to know that DCPIP is used to test for vitamin C, right? So we can note that this is a blue dye. We know it is blue in color. It will turn acidic. It will turn pink. Sorry, it will turn pink in acidic conditions. Right. Now the full name of it will turn pink in acidic conditions. Now you don't really have to know why it will turn pink in acidic condition, so I probably should not tell you about this, but it's good to know. The main point here is that it is blue. It is a blue dye, DCPIP. However, if vitamin C is present, basically the positive result, if vitamin C is present, we will see that the vitamin C, the color changes from blue, which is the original color, to colorless. So if vitamin C is present, it will change the color to colorless. Okay, so that's something that you have to note. This is one of the exceptions. Okay, now this is all for the test that you have to know. Second of all, in the syllabus it says that you also have to describe the structure of DNA. Okay. Now we have to know the structure of DNA. How does DNA molecule looks like? Now this is basically a very brief intro to DNA. This will be further expanded. This knowledge will be further expanded in your second year, and as you study more chapters. Now, for an overview of the structure, you have to know that DNA is actually stands for deoxyribonucleic acid. This stands for D-nucleic acid. Now, DNA is actually made up. It is made up of two strands coiled together to form a double helix. Now, note that I use very specific terminology here. Double helix, coiled strands. Now, DNA is basically two strands which are coiled together antiparallelly to form a double helix. Now, in these double strands, each strands contain chemicals called bases. Now, these bases are actually nitrogenous in nature, meaning they contain the atom nitrogen in the chemical nitrogenous base. Now, these bases actually determine which protein molecule does DNA have to produce. Now, DNA is a very important molecule because DNA actually is the mastermind of the cell. It is basically the commander, the captain of the cell, if you would like. So it actually controls cell activity. How does it control cell activity, Max? Now, DNA actually contains genetic information. Essentially, it contains the instructions to make proteins. It contains instructions to make proteins. Now, these proteins are really essential to life, because without proteins, your body would not be able to do anything. Now, proteins are these essential proteins such as enzymes. Now, enzymes add protein. hemoglobin, the substance, the chemical that is carrying your the protein that's carrying oxygen, right? They are all proteins. Your fingernails, your hair, they're all made of protein. Now DNA is actually the one who decides which protein to make or which proteins chooses to be expressed in various areas of the cell. Now, okay, I went a bit topic there, but next you have to know that each pairs or bases hold the strand together. Now these two strands are very specific strands together. You will have to know that there are 4 types of bases in DNA. Note that I say DNA, not RNA. So there are 4 bases. There's A, T, C, and G. Now A stands for adenine, T stands for thymine, C stands for cytosine, and G stands for guanine. Now the way that these bases pair up together is really specific. Now we call them, they are complementary to each other, complementary base pairing. Now you don't really have to know the exact reason or the mechanism behind how they pair and why they pair to each other, but you will only have to study that A will always pair with T, adenine will always pair with thymine, and cytosine will always pair with guanine. So remember this: A and T, C and G. Now in the exam, you will be likely to given a few different types of cells. Now and then they will ask you to basically find the complementary strand. Now the complementary strand basically A pairs with T, so T is gonna come. T pairs with A, G, C, T, A, T, G, C, C. Now this is the complementary strand. They would most likely ask you these kinds of questions. Right. Now that's all you have to know. However, I still want to conclude as a preview as a preview when you study genetics, you will have to just study the DNA molecule in great detail. Now, I just want to say that DNA molecule. Okay, so I think I probably should draw it out. DNA molecule. Now I know it's not the perfect drawing, but I hope you can see this. The bases are inside of the strands. You can see. Now this line thing here is actually a sugar phosphate backbone. So the chain in which the bases are connected is joined to is actually a sugar phosphate backbone, and the bases are nitrogenous bases, right? And then thirdly, we can see that this whole thing is called a nucleotide. Now that is one of the new vocabularies that you will learn, one of the new terms that you will learn. Right. So, and also one more important detail that I left out is that DNA is made up of sugar. Remember deoxyribose. It is called deoxyribonucleic acid because it is made out of deoxyribose sugar. Now, how do I know it is a sugar? If you forget that deoxyribose is a sugar, I would suggest you to look at the last 3 letters. Now it ends with O's. Now O's basically means sugar, right? So if the suffix or prefix ends with OSE, then this means it is a sugar. So glucose, there's an OSE. Fructose, there's an OSE. Sucrose, there's an OSE. Now these are all sugars. Now this is how sugars are named as well. So remember, deoxyribonucleic acid, DNA, is made out of deoxyribonucleic acid, not ribose sugar. And ribose sugar is another entirely different sugar which makes an entirely different molecule, which is not DNA. Now, this is everything that you have to know from chapter 4, biological molecules. And I hope you understand every single thing that I've try to explain. So, Thank you for watching once again. And if you have any questions, please leave them down in the comments. I will be more than happy to get back at you. Okay, until next time. Bye.